As you look. We are starting lecture unit 2. Lecture unit 2 has everything to do with neuronal physiology, but also digestive system, also skeletal muscle system. And how is it that I link all of those together in unit 2? They all rely on membrane structure and function and transport. Neurons are going to rely on membrane transport to generate an action potential. So does skeletal muscle. Your gut relies on membrane transport in order to absorb your nutrients. That's how they're all linked together. Also in lecture unit 2 will be a lecture on metabolism. Why is that important? Well, if you're absorbing nutrients, we should talk about how they are metabolized. Arguably, this is not the first time you've had a lecture on what the cell membrane looks like. It's not the first time you're going to hear about diffusion or facilitated diffusion. Arguably, you've probably heard about this now maybe 5 times in your life, starting in middle school, then high school, and then even the prerequisites here. I'm going to tell it to you all over again, but I'm now going to tell you why you care as a future healthcare provider. Because there are some disease states where it comes down to failure of membrane transport. Many of them that I can think of right now at the top of my head. The big one, diabetes mellitus. It is a problem with membrane transport. So we're going to go through it all over again just to make sure that you heard it right and now you know how to apply it for this entire unit. Up on the screen, you're seeing a phospholipid bilayer. You know that the phospholipids have a phosphate head, which is water-loving, and hydrophobic tails. This is not the first time you've heard of this, but we're going to go over the chemistry of those phospholipids today again. Embedded in the cell membrane, we can see proteins that span the entire membrane. Those are called integral. Or you can see proteins where they're only found on one side of the cell membrane. Those are called peripheral. There are other components found in the cell membrane, cholesterol, for example. We even can have sugars attached to either the phosphate heads or proteins. And this is also not the first time, with me at least, that you've heard about sugars that can be linked to cell membranes. You learned about that already, of course, for blood typing. So let's go through the proper membrane structure, identify what each of those components are going to do, and then we'll actually start talking about membrane transport so that you can apply it to all the systems that we're going to learn in Unit 2. What is the role of the membrane? Well, we can have a very exhaustive list, and then we would be here for 3 days. But its biggest role is going to be that it's semi-permeable, and it's selective. What does that mean in plain English? Some things get through, and others don't, at least not easily. That's what we mean by semi-permeable and selective. But we also know from our blood typing lecture that the other components of the membrane can be used to help identify self versus non-self. The sugars, for example, that are attached, the proteins that we see coming through the membrane. We think of Rh factor, for example. We also know that these proteins can provide a route for substances to travel through to get to one side of the cell, inside or out. We also know that these proteins that are found here can be used as enzymes. We know that they can be used to help link one cell to another. There are many roles that the membrane has, many roles. When we look at the phospholipid bilayer, we see the phosphate heads that are oriented to 2 different body compartments. They are water-loving. So one layer is going to face the extracellular fluid, and the other layer is going to face the intracellular fluid, what we call the cytoplasm. Attached to these phosphate heads, we can find other chemical molecules attached to it. Sugars we learned from our blood typing lecture, but also other water-loving elements can be found here. The phospholipid tails, the fatty acid tails, are linked to a molecule called glycerol. You don't need to worry about the molecular formula of glycerol. You don't need to memorize how many carbons it has and how many hydrogens are attached to that carbon. You should just right now know that glycerol is going to be seen again when we get to our metabolism lecture. Fatty acid tails are long hydrocarbon tails. They are not water loving, and that's why the fatty acid tails orient towards each other in the lipid bilayer. I like to think of this as a peanut butter and jelly sandwich, where the bread is on the outside and all the gooey deliciousness like peanut butter and jelly is the internal layer. The fatty acid tails create a zone of hydrophobicity. That means doesn't like water. And therefore, anything that does like water is going to have a hard time getting through that zone. In chemistry, you might remember something that your teacher said like this: like dissolves like. Like dissolves like, meaning chemicals that had similar chemistry like to associate with each other. Anything that's lipophilic, lipid-loving, is going to be attracted to this layer. Anything that is hydrophilic is going to be attracted to the body fluid compartments. You learned in your endocrine system that some hormones are steroids, lipid-derived, and you learned that they could get through the cell membrane very easily. Why? Because they are lipid-derived. They like this zone, this hydrophobic zone. When chemists look at the phospholipid, a single phospholipid, they see the water-loving phosphate head and the water-hating fatty acid tails. That makes a phospholipid a combination of both. And terms that describe that is amphipathic or amphiphilic. You've heard of amphibian. That means a creature that loves both water and land. Well, amphipathic or amphiphilic describes a molecule that is both water-loving and water-hating. Any questions about this? Let's talk about the proteins that can be found within the cell membrane. As I said before, some can actually span the entire cell membrane. Those are called integral proteins. Some are associated only with one side. Those are called peripheral proteins. What do these proteins do? They can be important for cell-to-cell adhesion. They can be important for knowing self versus non-self, so cell recognition. They can be important channels going through the cell membrane. These channels can have gates to them. If the gates are closed, then a substance can't get through the channel. That's what I'm showing you over here. How do proteins form gates? Well, their amino acids loop out and can block the lumen of the channel. Something's going to have to happen to make the amino acids shift, make a conformation change to open that channel before something can go through it. We will address that somewhat today, but in great detail in the future. Some channels are not gated, in which case they are called pores, meaning they are a constant open channel going through the cell membrane. A great example of that is something called aquaporin. Well, what does aqua mean? Water, good. And porin means pore. So put it together. It's a pore to allow water to go through the cell membrane. I actually was on an interviewing someone to come teach here. My partner in crime. I was interviewing for my partner in crime, teaching physiology. And this guy, we asked him, what are your strengths as a professor? He said, well, there's a lot of language in physiology. And I like to teach them their prefixes and suffixes so that they, through just knowing a prefix and suffix, they understand better a new term I might tell them. Like you all now know zymogen, ogen, right? Fibrinogen. And you also know prothrombin. You know that all of those tell you it's a protein that has to be activated, right? So he's talking about how he really focuses on the vocabulary, and I'm getting all excited thinking, this is the one. This is going to be my work husband. This is exactly an issue. I'm so proud. And then we ask him, could you please tell us the different kind of urine that a person would make if ADH is released, antidiuretic hormone, leading to aquaporin insertion? And this is what he said. Could you please tell me what an aquaporin is? Now, is the irony lost on you? That he just told me how he spends time on prefixes and suffixes, and he couldn't figure out what that word was. You all just did. I said, what does aqua mean? You went, water. Porin, pour for water, right? I haven't even taught it to you specifically and what it does in your kidneys, but here's this physiology teacher who's taught it before, and he's like, could you tell me what an aquaporin is? And then I came, my dreams and my visions just went, yeah, he did not get hired. Some proteins are going to shuttle substances across. They're going to carry substances across, and we'll talk about that in just a moment. So you need to keep track of the sidedness of the cell membrane. Remember, we already talked about this in our body fluid compartments lecture. We find different substances. Is everyone in the class right now? Everyone's here, right? So would you mind closing the door, because it's getting loud? But the door will lock, so if you leave, put the trash can back out by the door so you can get back in. Okay, that's much better. The sidedness of the cell membrane is important for us to remember. Let's review. Where do we find more sodium? Outside the cell. Where do we find more potassium? Inside the cell. Good. Where do we find more proteins? Inside the cell, where it's made, right? Can there be a body fluid compartment that is protein rich, though, that's not inside? You learned about plasma being rich in albumin, for example. So that's what I mean by the sidedness of the cell. What else can we find? Carbohydrates linked to the phosphate heads, or even to the proteins, or just hanging out in the extracellular interstitial fluid. These proteins are glycoproteins and glycolipids. And altogether, this gives a nice little sugary outside coat to a cell called the glycocalyx. Now, these sugars typically have a negative charge to them, and you're seeing negative signs up on the screen. But what I need you to know is it's all relative. If I ask you to compare the negativity of the outside of the cell with the glycocalyx to the charge inside the cell, you will learn this week and next, in the next couple of weeks, that the inside of the cell is much more negative than the outside. So don't let this mislead you. Yes, the glycocalyx is slightly negative, but it's not as negative as the inside of the cell. I will tell you more about that in the coming days, but not today, my young Padawan learners. It takes time to develop this concept. We know that those sugars can play a role in the immune response. We learned that, again, with our blood typing lecture. Cholesterol is also present, associated with cell membranes. Cholesterol helps augment the hydrophobicity of this internal layer. Cholesterol acts also as an antifreeze, preventing cell membranes from freezing when exposed to cool temperatures, cold temperatures. It also gives more support to the cell membrane. Let's talk about transport through the cell membrane. When scientists look at a cell membrane, when they study movement through some sort of membrane, dialysis membrane, cell membrane, they ask questions like, what can get through? And if you already did your PhysioX that I asked you to do for Thursday, you were assessing what could get through on its own. There was an experiment on simple diffusion. And we will talk about that on Thursday after your exam. Hydrophobic molecules, like your gases that are dissolved in your body fluid compartments, can get through the cell membrane on their own. No help required. Simple diffusion. If we look at small, uncharged, or slightly polar molecules like water, it can get through the cell membrane. It doesn't have to go through aquaporins. If we look at bulky molecules that are uncharged but polar, their size becomes a restricted issue for them. They can't get through as easily. But what about small charged ions? They can't get through either, even though they're so small. So that's when we're looking at a non-living membrane, like a dialysis membrane. But when we actually look at a living membrane, we get very different results for some of those items I just went through. Gases, they still get through, no problem. But the ions, the water, glucose, which is very bulky, they get through too. And the question is, well, what does a living membrane have that a dialysis membrane doesn't? And the answer is proteins. It's proteins. We will be playing with a dialysis membrane next week for a wet lab in class. So you get to have fun again next week. After all your exam is over, you get to have fun again. We're going to do a dialysis membrane experiment using sugar, and we're going to see when can it get through and when can't it. Which side does it migrate toward, if at all? You've probably done something like this many times. How many of you have taken Bio 20? If you did, you certainly did a membrane transport experiment. So it's an easy experiment, but at least something fun that you get to do. So living cell, we have proteins. Now we're talking about membrane transport. Passive transport simply means that you do not require cellular energy like ATP. And look, energy comes in different forms. I'm saying cellular energy, ATP. Passive transport has two smaller subdivisions, simple diffusion and facilitated. We're going to go through each of these individually. But how are they common? They allow a substance to go down its concentration gradient. Does everybody know what a concentration gradient is? What is a grade? If you're coming down a mountain or going up a mountain, aren't there usually signs to alert the truckers about the percent grade of the incline? Especially if they're going down, say, the grapevine. If it's too steep of a grade, they gain too much momentum, their speed accelerates. The trucker tries to put on the brakes and their brakes can fail. Have you ever seen the emergency braking platform of rocks and barrels to stop them safely. So that's a grade, a steepness, slope in math. They called it slope, not grade. So a gradient for passive transport. You've learned that things go from high down to low, right? How many times have you heard that? From a high concentration gradient down to a low. Some passive transport, there will be a carrier or a channel, but simple diffusion doesn't always need a carrier or a channel. In this picture right here, it's showing you a channel. And in this picture right here, they're trying to show you a carrier. I'll show you a better example of that in just a moment. No additional energy is required, just the energy of movement of that substance. Because remember, molecules do have movement from the bonds that they have and the electrons in those bonds creating vibrations of the molecule. So they're moving. That's kinetic movement, but no cellular energy is required. In striking contrast, active transport, we're going to have a special protein. It still is going to act as a carrier, but it's going to move at least one substance, sometimes more, against their gradient, pushing it up the mountain, if you will. Well, that's going to require energy. Definitely will. How much gas do you consume when you're going up the mountain? How much gas do you consume when you're coming down the mountain? You might not even need to press on the gas pedal, right? I drive a car where it's electric. When I go up the mountain, I see my electrical storage in my car drop. But when I go back down the other side, the car's battery has the ability to retrieve that kinetic energy, that going downhill, and it will recharge my battery. So I kind of get, not all of it back, but a lot of it back into my battery. It's kind of cool the way it does that, yeah. Yes, thank you, Elon Musk. This is going to require ATP if we're moving something against the gradient. But there are two different kinds of active transport that fall in this category. Primary active transport, and one that's a little bit more quirky called secondary active transport. Again, I'm going to go through all of these one at a time. Let's review a special kind of movement called osmosis. We've already talked about this. We've already had a chemistry test on it, but let's review. If we find unequal particles on either side of a cell membrane per unit volume, if the particles cannot diffuse, what will move? Water. Water will go from high water concentration to low, but another way of saying that, same coin, other side of the coin, is water will go to where there are too many particles. Particles suck, we learned. If cells lose water, they crenate. If the cells gain water, they can rupture. We would like cells to be in an isotonic solution. Our magic number, remember, is 300 milliosmolal. The net movement of water from one side to the next is what we are saying when we are using the word osmosis. Water is moving from one side of the cell to the outside to the inside all the time, but when there's a net shifting of the body fluid compartments, that's osmosis. And we've already had our body fluid compartments where we appreciate how quickly water can shift. Again, next week, you're going to do a lab experiment where I will prove to you how quickly the water can shift. Here's another picture showing you osmosis. Here is our selectively permeable membrane. On one side, we see a lot more particles than the other. If the particles can't move, we recognize that water will move toward them in an effort to dilute them. The Physio-X that I have assigned for Thursday, you're going to be using a manometer tube system. How many of you have done it already? right? You use this U-tube kind of apparatus, and you had a membrane in the middle, and you were using a piston trying to measure how hard you had to push on the piston to stop the water moving from one side to the other. You were using hydrostatic pressure indirectly to measure osmotic pressure. The more particles there were, the harder you had to push to stop the water from moving if the particles couldn't. And the PhysioX does a really good job of giving you examples of how hard you have to push when the particles can't move versus how you didn't have to push if the particles could move. When you do that experiment, please pay close attention to the concentration you were playing with and the type of solute you were playing with. Because you know why? Dissociation factor, something that we work so hard to understand, is definitely being shown to you in those PhysioX experiments. They get gold star to PhysioX on this one. They get everything right, and it's a good experiment to do. You were tested on something like this on your chemistry exam. We have solute A that has a molecular weight of 100, and I put 100 grams in a liter of water. So this is a 1 molar solution for solute A. Solute B has a molecular weight of 1,000 grams, and I put 1,000 grams of it in 1 liter of solution. This also is a 1 molar solution. Some of you forgot that moles per liter, if everything is 1 mole per liter, then they have exactly the same number of molecules. 6.02 times 10 to the 23. But if something dissociates, well, that changes it. Then they have different osmolality, potentially. That's where we had to worry about the dissociation factor. If solute A and B don't dissociate, well, then they would have the same osmolality in this experiment. But if solute A had a dissociation factor of 3, then we would say it was a 3 osmolar solution. So osmolarity is an index of the particles. Molarity is an index of the molecules. Dissociation factor distinguishes between the two. Simple diffusion, part of passive diffusion. This is where particles are allowed to get through the membrane and they move a net movement from high concentration to low. I'm using this slide to represent a gradient. Now, when my boys were younger, simple diffusion happened real powerfully one day. And it was a day when the boy, again, they were young. Mac was just being potty trained, and Hayden was already potty trained. And one of the things that kids are really good at, and I will teach you this in lecture unit 2, is they obey the law of the gut. There is a real thing called the law of the gut. And simply put, it means food in, poo out within about 20 minutes. All of you, did you go outside and have a snack during our break? Did you eat something somewhat substantial to fill up your tummy? No? Some of you are going, yes. Did you have breakfast this morning? Did you poo as a result of it? You haven't yet? Because you're not obeying the law of the gut. Some of us are better at it than others. But 20 minutes of putting food in your mouth, you will have fecal material, not from what you just ate, but from a previous meal. You will have gut motility that will move more fecal material into your sigmoid colon. And just as much as 20% full, signals are sent up to your brain going, you could poo. How many of you obey that feeling? I do. Heck, I just need to take my morning medication, a tiny little pill, with some coffee, and it's fire in the hole. 20 minutes, fire in the hole. I can count on it. I obey my law of the gut. And I'd like to get that over with before I come to class. I like my bathroom. But sometimes it doesn't happen until I'm arriving on campus, and then I pull over somewhere else to my secret bathrooms where it's sanctuary. No one is in them at all. At all. It's just quiet. I can hear birds chirping. It's awesome. I'm not telling you where my secret chambers are. And they are not faculty either. They're for students. But kids obey the law of the gut. 20 minutes of feeding them, if they're at the table, guaranteed. Where are my parents in here? You're at a restaurant. They bring the food to the kid first to keep them happy. But what they don't know, these waiters and waitresses, is the law of the gut. Because they are done eating, you're just now getting your food, and that's 20 minutes later, and now you've got your kid going, I'm gonna go to the bathroom. And then you Rochambeau who has to go take the kid. As a parent, you don't ever have a warm meal anymore because someone's going off to take the kid to the bathroom. Am I lying?
That's true.
So the boys were at the dining room table. Hayden says, law of the gut. He's like, Mommy, I gotta go big potty. It's like, okay. I get him down off his big chair, and Mac, being a toddler, it was always, my two, Mommy, my two. Like, okay, you go too. And so they go off together, and Mac would sit in his little potty training chair, and Hayden would be on the big potty, and they would be in there laughing, telling stories, singing, forever. And they're still that way. They can spend hours in the bathroom. I don't ask what they're doing at this point. And to a parent, as long as they can hear noise, we think, well, they're okay. So my mom was over, and we're having a nice conversation because the kids, we can hear them, they're happy, and it's gonna be a while. And then all of a sudden, the smell hit us. And it was a really horrible smell. Sort of smelled like a French whore. A lot of perfume. But nasty mixture of perfume. And so Mom and I went racing down through the dining room, to the hall, to the bathroom door. Boys, I go to open it, locked. Boys, unlock the door. Not yet, Mommy, we're making a surprise for you. I was like, no, that's okay. No surprises. Just unlock the door. No, we're not done yet. So I go and I get a screwdriver and I pick the door lock and I open it. And it wasn't that they did anything with their feces. I know what you're thinking. But we were getting ready to move, and I had all of my perfume bottles in a crate. And they decided it would be a good idea, because they saw Honey and I painting, that they would paint the walls with the perfume to leave it smelling good for the new people who are moving in. Some of those perfumes actually remove paint. So they were painting, it was just, so now think about this diffusion. They were in an enclosed room, door was closed, window was closed, high concentration of fumes. On the outside of the bathroom door, low concentration of fumes. And there was permeability. Where? The crack of the door. And when we talk about the rate of diffusion, how quickly it happens, there are several things that will increase the rate. One is have a higher gradient. How fast is this kid going to come down the slide if I made it steeper? If I made it less steep? Slower, right? So was there a high concentration gradient in the bathroom? Absolutely. Was there a low on the other side? Absolutely. Was there permeability? Yep. And those fumes came out from under the door, banked down the hall, through the kitchen, to the dining room, and it was like, hits my mom and I in the face. Pretty quickly. That's why we were alerted that something bad was going down. That's my pooping and peeing story and perfume. I'm going to try and write a book that's on my bucket list. A to Z stories of a Z to A family, something stupid. I'll think of a better title. But that's my P chapter. My letter P for pooping and perfume. Mm-hmm. Now, can a molecule move from this side back outside? Could it? Could something move against the gradient? Isn't there random motion in those bonds that I was telling you about? But overall, when we speak of diffusion, it's a net movement. Overall, which way will the substances move in this picture? From outside to in. So the random molecule that worms its way against the gradient, I like to think of that idiot when you are on an elevator, a crowded elevator. Don't you have high concentration in that elevator? Do you have permeability if the doors are closed? No. When the doors open, from now on, when you're on an elevator, you're going to have this thought in your head. When the elevator doors open, you now have permeability, and you would prefer to diffuse out of the elevator to the floor that you want to go to. There's always that moron who's on the other side who tries to get on the elevator before letting everyone off. Why is that annoying? Because then you're doing this, right? Next time you see that person trying so rudely to get on before you all get off, look to your neighbor, go, you got him? Okay, let's go. Pick that person up by their elbows, walk them back outside and go, no! Fix law! And then walk away. Maybe they'll learn. Fix law. Simple diffusion comes in many forms when we're talking about a biological membrane. Feel free to ooh and ah as I click through this because it takes a long time for me to do this. Ready? Molecules can just move. It's like July 4th with the fireworks. Like, ooh, ha, ha. So molecules can move through the cell membrane with no assistance whatsoever using simple diffusion if they're permeable through that hydrophobic zone, like CO2 or oxygen or nitrogen. Some substances go through a channel that's open all the time called a pore. Some substances want to move through a channel, but if it's gated, they can't go. Once the gate is open, then they can. All of those are examples of simple diffusion. The proteins that are there in these last two examples merely create the channel. There is no physical binding of the protein to the substance that's moving. There is just merely a lumen that they have created. As long as the lumen is open, a substance is allowed to move. Now, these channels we are going to learn in our Unit 2 are going to allow specific substances to go through. Even if it's a pore that is open all the time, you might think anything can go through. No. These channels are going to still be specific for some entity, some molecule, some ion to get through. Aquaporins. They're called aqua because what do they allow to move? Water. Even if it's a channel that's open all the time, the amino acids that line that lumen are going to give specificity. So that means scientists have to name these channels, these pores. Aquaporins tells you water gets to go through. Pore meaning it's a channel that's open all the time. But if I say sodium channel, guess what gets to go through that? Sodium. Calcium channel, calcium. Again, the amino acids dictate what can get through. There's size exclusions as well. How big is the channel? So size, but also the chemistry. Sodium has different chemistry than calcium. Calcium has different chemistry than chloride. And that becomes important to us when we learn about neuron and skeletal muscle physiology. Now, some of these channels are gated, and something has to open them, trigger them to open. Where are your ligands? Where are they? Where are your ligands?
My bag.
In your bag? Ligands. We talked about this, right? Your keys. So how do you use your key? Don't you insert it into some sort of orifice? Right? Or here's my door key. I have to swipe it in front of some sort of lock. But all of these are ligands. They have to interact with some sort of lock in a way. So a ligand-gated channel, in biology, a ligand is some sort of chemical. We've already learned about this. We learned about hormones being ligands to bind to receptors. Ligand-gated channel. From now on, when you use your key to get into your front door, you're going to say to yourself, I'm using my ligand to open my ligand-gated channel, your door. And now I'm allowed to diffuse into my home. You have to go through all of that. Announce it too when you come in. Hey everyone, I used my ligand to get through the ligand-gated channel. I've diffused home now. Aren't you glad to see me? And they're going to say, what did you learn at school today? But when I get home at night, I don't use my ligands to get into my ligand-gated channel of my house. I instead put my car in the garage. And I have to open my garage door in a very different way. I push a button, and a signal is sent from my button inside my car to the transponder inside my garage, and the garage door opens, right? I have an automatic door opener, garage door opener. And I like to see how far down the street I can be where I can push that button to see how far I can be to get the door to start opening so that I can just smoothly drive right in without having to stop to wait. And then as I'm pulling in, I hit the button again so it closes. I like to think that I'm sneaking home before the blue hairs can actually tally. Oh, she's home now. I like to do that. But sometimes the battery goes dead. And then what happens? I can't open my garage door. So the batteries, I don't know if you know this, but they have different values associated with them. 9-volt battery, AAA batteries, AA, D batteries, C batteries. I don't know what happened to B. But all of them have different sizes and they have different chemical storage inside. And they have different volts. Some are more powerful than others. So a voltage-gated channel is going to be something where something chemical, something charged, has to change across the cell membrane to get it to open. So for me, when I get home and I'm opening my garage door, I'm like, honey, I'm home. I used my voltage-gated channel to get in the house today. That's my garage door opener and my garage door. My garage door opener is the volt change. It has a battery. It sends the signal if the battery is working, and then the gated channel opens. So what does that look like visually? If we have separation of charges that change in a certain way, and that's the part I have to teach you, not today, but if they change, the voltage gated channel can open. Don't batteries have a negative pole and a positive pole? Okay. So we're going to talk about this polarity, this charge difference across the cell membrane. If that charge changes, voltage-gated channels can open, and then something can travel through it. We just got to learn what triggers it to open exactly, how is it triggered, and what will travel through. A ligand-gated channel. Your keys. Here's my little blue pentagon. That's going to bind to the receptor and open that channel. In Unit 2, we call those neurotransmitters. They're going to open ligand-gated channels. We'll learn what neurotransmitters are. But again, we could have a channel that isn't gated, in which case something is just allowed to move through it. All of these are examples of simple diffusion, and I promise you, you are not listening to me right now. Statistically speaking, 20% of you are not listening, even though I said you're not listening right now. And in your head, you're like, yes, I am, because you said. And now you're instantly in la la land again. You're like, my daydream was a lot better than this shit. And on lecture exam 2, is lecture exam 2 even on your radar yet? Because it is for me. I'm starting it today, and I know what I'm going to put on that test. I'm going to ask you to match descriptions of different kinds of channels to what kind of movement they allow across a membrane. And I promise you, I'm going to put down there sodium voltage gated channels. And you all will select the wrong answer, even though it clearly states simple diffusion. Don't make that mistake, because statistically speaking, the 20% of you who aren't really paying attention right now will forget we had this conversation. You'll get your lecture 2 exam back and you'll say, can you please explain to me why sodium voltage gated channels aren't active transport? Why is it simple diffusion? And I'll go back and watch the YouTube video where I have it documented, because I'm recording it today, where I said, you're not listening, because that was you. Simple diffusion. Once the channel is open, there is no physical binding of the protein to the solute moving. It's just merely going down its concentration gradient once it's allowed to move. Once I figured out how to do all that movement, I did too much of it. There we go. I'm going to keep going. How did we learn about these gated channels? Well, there was a technique called patch clamp technology. The two people who developed this actually did earn the Nobel Prize in 1991. You don't need to remember their names. But simply put, this is what they did. They took, I'll use something that you can visualize. How many of you in chemistry or biology courses, you've used those Pasteur glass pipettes that have a long, narrow neck, and you would put a little bulb squeegee thing at the end, a second squirt. Well, in my lab, we would take those Pasteur pipettes and we would hold them over a flame. And over a flame, glass will melt. So that long, narrow neck would start to loop as the glass would melt. And we would pull as soon as we saw that happen. And what that allowed us to do was, as we pulled, we were able to take that long neck and make it become more and more slender with a smaller and smaller lumen, to the point where we could then take those Pasteur pipettes, attach them to a vacuum, and we could actually, under a microscope, we would have a petri dish of cells, freshly fertilized oocytes, and we could use our vacuum with our Pasteur pipette, and we could go, and we could actually suction ever so gently a cell to our Pasteur pipette, hold it stable. And with another pipette, very finely, like a needle, On another pipette, we'd come in on the opposite side and inject a gene into these oocytes. So these scientists did a similar thing. They had this Pasteur pipette. They sucked a cell, stabilized it, and what they did was just added different chemicals to the solution. And they, with a different measuring device, an electrode, they were able to detect if the gate opens and ions flowed. If the ions flowed, they would be able to detect that on a voltmeter. They can actually see an electrical current. So by random chance, they would have the cell sucked up. There would be proteins that would happen to be within that region of the glass pipette holding it there. And they could add different chemicals to find out if it opened ligand-gated channels. Depending on the chemical, that's how they named the ligand-gated channel. For example, nicotinic acetylcholine receptors, abbreviated like this. That is a kind of ligand-gated channel. It's a receptor that has to bind to a ligand, and when it does, it opens. What triggers it to open? What is its ligand? They named it for the ligand that is normally responsible for that, acetylcholine. But they tried other drugs as well, and they found out that some of these receptors that they discovered would open if they added nicotine to the solution. And so they named it after both, nicotinic acetylcholine receptors. You will find in Unit 2 that these ligand-gated channels can be named for how they work. They can be named for the chemical that opens them. They can also be named from some sort of drug or toxin that can also open them. There's another kind of receptor they discovered, muscarinic acetylcholine receptors. Well, acetylcholine also opens that variety, but so did a mushroom poison called muscarine. And that was an important discovery because do you randomly go around and pick mushrooms and eat them? Why is that dangerous? Inherently, you know why. They're poisonous. Well, what will you look like if you eat the poisonous ones? Because chances are you're going to eat one that has the muscarine toxin. And that means anywhere in your body where you have that receptor where muscarine can open it, it will. Where do we find it? In your gut. Where else do we find it? Your lacrimal glands. Where else do we find it? Sweat glands. Where else do we find it? Salivary glands. You eat that toxin, it's going to go everywhere in your body. You're going to have oozing from every pore and orifice. You're going to be crying, salivating, sweating, increased gut motility, diarrhea is what that's called. That's a horrible way to go. Horrible way to go. And every year, if you pay attention to it closely enough, there are reports, news reports, media reports of families that went out to pick mushrooms and they got their identities mixed up from their picture book and they picked the poisonous kind and it wipes out families. Yeah. Not too long ago, some little kids died from it because the mom gave them mushrooms that someone picked for her. They got the identities wrong and her kids died. It's horrible. Bad way to go. So they discovered lots of different channels, the voltage kind, the ligand kind, using this technology. Now, facilitated diffusion is still considered to be passive transport. But facilitated diffusion is different in some ways than simple. Facilitated diffusion is like simple diffusion in that it does not require cellular energy. Something is moved from high to low, a net movement from high to low. This movement, like simple diffusion, will continue to keep going until there is no gradient left. We call that equilibrium. But how is it different? There is a physical interaction of the protein with the solute it is moving, a physical binding. And that means we're going to have a rate limiting factor. You want more things to be moved this way? The rate limiting factor is the number of carriers you have. You want more of the solute moved? Then you need more of these proteins made. And that means more gene expression. This means we're going to have something that's called transport maximum. When we're speaking of how rapidly things move across a membrane, we use velocity maximum. But when you see a T max, you immediately switch over to, oh, there's some sort of protein now involved, transporting this solute. I could open the door and say, you all are free to diffuse home now. Class is over. And you might be looking at the clock saying, Kara, it's only 10 after 11. We don't finish until 12:30. I don't feel comfortable with this. Students who leave early usually lose out some way. I'm not leaving. I say, no, really, I'm done. This is a good stopping point. I want you to get home early to go study. Some of you still might be hesitant. Tabitha says, I'm not going anywhere. Well, I could physically come over to her, grab her by the arm gently, and escort her through the door saying, no, really, it's time to go. I have a meeting that I have scheduled. I'm letting you out early. I am physically moving her out the door. Maybe the rest of you say, I don't trust her. I could come to each one of you one at a time and escort you out. There are 30 students in this class. If it takes me 1 minute for every student to escort you out the doors, the very last person here is going to be waiting 30 minutes. Agreed? So it will take me 30 minutes to transport you all out of this room. That's my transport maximum. There's only one of me. But what if I got 6 other teachers in here to help me with that? Won't we transport you out in less time? So anytime a protein physically interacts with a solute it's moving, there will be a transport maximum. The rate limiting factor is the number of carriers you have. You want faster transport, you need more carriers. And diabetics have increased blood glucose levels, but that glucose also comes out in their urine. Glucose is freely filtered by the kidneys through the glomerulus into the renal tubules. The renal tubule cells have carriers whose job it is to grab the glucose from the filtrate and put it back in the blood. But if too much glucose gets filtered and you overwhelm those carriers, then some of the glucose doesn't get moved back into the blood and it ends up in the patient's urine. Finding glucoseuria, finding glucose in your urine, your patient's urine, is a problem with facilitated diffusion. You don't have enough carriers to keep up with the extra glucose that's been filtered. Now, in a PhysioX that you will do eventually, you actually build more carriers into the membrane. But physiologically, a diabetic can't do that. The cells of the kidneys have no way of going, oh wow, there's more glucose in the filtrate than normal, more gene expression, insert more carriers. They don't have that ability to do that. It doesn't exist. So some of the glucose is on its way to the toilet. Glucoseuria is a finding in uncontrolled diabetes. Let me use this example. Here you have glucose in a patient's filtrate. It's on its way to the toilet unless those carriers can grab it and put it back in the blood. The problem is they can only work so fast. They have to bind the glucose, they have to move it across the cell membrane to the other side. They let go, then they have to change their conformation to come back for another one. And they only can do this so fast. Well, meanwhile, glucose in the filtrate, it's like, I'll just hang around and wait for you to come back for me. No, the filtrate keeps flowing. So if all the carriers are bound, whatever glucose didn't get bound is on its way to the toilet. It can't just hang around waiting for the transporters to morph back over and say, I'm open now, I'm an open taxi, I'm available. That's what we mean by transport maximum, rate limiting factor. So velocity maximum refers to processes that have simple diffusion. The greater the concentration gradient, if they have permeability, the faster they move. That's the boys with the perfume example. But if you have facilitated diffusion, the ability to move the solutes becomes saturated. All of the transporters are saturated, occupied. They can only move back and forth so quickly. And that means that's the rate limiting factor. They will max out. Shown here in red is the transport maximum. In a PhysioX, like I said, you're going to play with that concept by building more transporters to get faster movement. Now, what about active transport? This is going to require, in one type called primary active transport, cellular energy, ATP. Secondary active transport also uses or requires ATP, but indirectly, not directly. And I'll explain that in just a moment. Let's first talk about primary active transport. And again, this is now a substance that's going to be moved against its gradient. The best example of that is primary active transport. The best example of primary active transport, I should say, is the sodium-potassium ATPase. Sodium-potassium ATPase removes 3 sodium out of the cell against its gradient. Remember, sodium is found abundantly outside the cell. And 2 potassium are pushed into the cell against its gradient. We've got 5 ions moving in different directions against their gradients. This is going to consume a lot of ATP. In some cells, up to 90% of the ATP made by the mitochondria can be used for just the sodium-potassium ATPase. It is hugely important. It's important for proper neuronal function, muscle function, renal function. And that's why I had that crippling thought when my dog accidentally got into that kind of rat poison. This is still carrier-based, which means you're going to have a transport maximum. The PhysioX you're doing, the very last activity, deals with the sodium-potassium ATPase. You might think that ATP is the limiting factor, but really, at the end of the day, if ATP is supplied as much as this channel needs, this protein needs, the rate limiting factor will be the number of pumps you have. That's the rate limiting transport maximum factor. Now, what about secondary active transport? Let me go back just a moment. Why do I have a picture of people skiing for active transport? So, secondary active transport does not directly use ATP. It indirectly uses it. So here's my stupid analogy. Who here likes to go skiing or snowboarding? Okay. So, Miguel, you raised your hand?
Oh, yes.
Okay. What do you like to do, skiing or snowboarding or both?
Snowboard.
Okay. I do not have that talent. I like to ski, but I do not have the snowboarding talent. So, I have this idea. Let's go skiing, snowboarding together, okay? But I hate sometimes ski resorts, they get really long lines. And I got a plan, because I'm a genius. And here's my plan. I have a really long rope. I'm gonna leave you at the bottom of the mountain. You're gonna tie yourself into that rope somewhere in the middle, okay? I'm going to take the other side of the rope and I'm going to take the ski lift chair. Doesn't the ski lift chair require energy to bring me up the mountain against the gradient? Okay. When I'm up there and I get off, I'm going to tie the rope around me. And I'm also going to loop it around the base of the ski lift chair, you know, the big pole. Miguel has done the same thing down at the bottom. I'm going to throw down the rope to him all the way down the mountain. I've got a good arm for it. And he's going to connect the two ropes together so that this is what we're going to look like. Here's the ski lift chair, machinery. Here's the ski lifts. I'm up here. Miguel's down here. We got the rope looped around us and around the base. And I'm going to ski down the hill. And as I ski down the hill, it'll pull him up. Could work. And then that way, number one, we don't fall. Number two, we don't have to wait in line. It could work. So the momentum of me going down the mountain is going to provide the kinetic energy to pull Miguel up the mountain. I'm going down a grade and he's going up. Genius. But in order for me to set up that system, I had to initially use the chairlift, didn't I? So there's an initial investment of ATP. But once the system is set up, you don't need any more further contribution of ATP. Now, some substances, in my example, they don't go in opposite direction like Miguel and I would be going. Me going down the mountain, him going up, then him going down the mountain, then me going up the mountain. These secondary active transport proteins can actually move substances in the same direction. But still, one substance is coming down its gradient and the other substance is going against its gradient. So they can move in the same direction across the cell membrane, but one is still coming down the mountain while one is being pushed up the mountain. These are called co-transporters. Now, the example that you see up there is sodium. Remember, I just told you about the sodium-potassium ATPase, which pushes 3 sodium out for every 2 potassium that comes in. You all told me that sodium is most abundantly found outside the cell. Well, what put it there? The ATPase. That's me using the ski lift chair to get up the mountain. If there is a totally different protein that allows sodium to come in the cell, sodium's going to go down its gradient, won't it? Diffuse back into the cell. That protein can also use that kinetic energy to pull something else into the cell, like glucose, against its gradient. That actual protein that moved sodium and glucose did not directly use the ATP. But ATP had to be used at some point in the system to establish the sodium gradient. Secondary active transport does not directly use ATP. We require the primary active transport to set up the system so that a different protein can channel into that kinetic energy. Now this picture shows countertransport or antiports. This goes better with my story of Miguel and I going skiing/snowboarding. Something's allowed to diffuse down its gradient, in this case sodium, while something else is pulled against its gradient. In this case, the solutes are going in a different direction across the cell membrane. Okay, let me stop my video.